Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Icahn. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@epin.info. My guest today is Professor Giulio Pasinetti, who's a professor of neurology, neuroscience, and geriatrics at the Icahn School of Medicine at Mount Sinai. He also serves as a director of basic and biomedical research in the Center for Geriatric Research and Training at the Bronx Veterans Affairs Medical Center. He is also the director of the Center for Molecular Integrative Neuroresilience at Mount Sinai. Welcome, Giulio.
Welcome. Thank you very much, Jill.
Sure. Yeah. So your lab does a lot of work in Alzheimer's and Parkinson's disease. And one of your primary goals of the lab is to investigate the biological processes which occur when during aging subjects with normal cognitive function convert into the very earliest stages of Alzheimer's and then progressively to dementia. You have determined that certain mechanisms may be at play early in the onset of AD, even before overt signs of symptoms. You want to talk a bit about what the lab is doing and what your focus is there?
Yes, thank you very much, Jill. I think I will, before answering your question, I think it will be very good just to give a broader perspective from where we are coming from and why we've been doing this work and why also NIH and NCCIH has been funding this program in a mechanism we call neuroresilience, but actually in promotion of resilience and the promotion of the resilience is always have to be or something that we could delay or we might eventually to prevent.
Yeah.
So the bottom line is that the program has been developed with the support of NIH, in particular the National Center for Complementary Integrative Health, as well as also the Office of Dietary Supplement and the National Institute of Aging, and is based primarily on just a very simple concept that in any disease, we are very interested in neurodegeneration, but any kind of degenerative condition that is characterized to be progressive, irreversible, and degenerative, is in some way something that research and science has been focused for two main kind of potential application. One is basically to try to have a disease-modifying strategy, basically to block the disease. The other one actually something that is maybe symptomatic. Both of these two appears to be in some way, well, the symptomatic approach for treating the sample, treating the disease appears to be in some way good, but in a very temporary manner because the disease actually progresses, is reversible and deteriorating. The other major alternative is actually to try to do disease-modifying strategy, basically to block the curve or shift the curve in a way that actually will slow down the progression or maybe attenuate the disease. So
the research projects in the Botanical Center to understand mechanistically the potential role of certain botanical dietary supplements, you say, in particular polyphenols. And the three different projects going on to promote resilience against psychological stress. That's Project One. Sleep disorder induced cognitive impairment. That is Project Two. And then clarifying the role of the microbiome. And that is Project Three. You want to talk a bit about all three of those things?
Sure. So about 4 years ago, I participated into what is being called the Lancet Commission for Prevention of Dementia. Actually, initially it was a Lancet Commission for Prevention and Treatment, and then it became the Lancet Commission for attenuation of the progression and prevention of the disease. So, in other words, what this commission was able to come up, and actually was the result of this kind of very fascinating kind of journey of understanding where we are coming from in terms of age and age-related disorder. We came up with this idea that about 65% of factors are basically not preventable. And most of those are associated to genetic kind of predisposition. So, but there was something very extremely interesting, and actually we are witnessing now, as I said before, some of these studies were actually demonstrated, for example, that with the mind diet, you can attenuate the mechanism of Alzheimer's disease or onset of dementia. This kind of other 35 potential preventable factors are associated primarily, as I said, to diet, are associated, for example, to many factors, like, for example, well, we know that the traumatic brain injury, for example, is one of the major risk factors in onset of dementia. But it's also true that we need also to keep in consideration that there are other kind of social aspects that like loneliness and depression is actually one of the most important things that actually to keep in mind. Anxiety, all of these factors that is in some way, they were still in part, let's say until a few years ago, in part disregarded as just in terms of causality. Because a very important thing is to be scientists, there's always to be sure they're able to discern what is maybe as an observational condition that it may be in parallel versus something that have a causality. Because if you don't have a causality, there is no an effect, and there is not a causal effect relationship, and there is no way that you actually can interfere with that. So actually, in the last 3 years, it became very clear that some kind of a mood disorder, like anxiety, depression, major depression disorder, PTSD, some of these kind of circumstances may play an important role into the mechanism of onset of dementia, stressful condition. So actually what we decided is in part to initiate some studies, and we did, like in a rigorous drug discovery screening, the identification tried to characterize several type of what we call polyphenol metabolites. Like actually we have done a pharmacokinetic study. Basically, actually we developed a study like in animal model where we develop a certain kind of dietary polyphenolic preparation. And then through bioavailability study, we were able to identify, first of all, which are the metabolites that can maybe just circulate in blood. Those were actually excreted through urine, or those ones that actually are capable to reach the brain.
Right.
Typical same study that actually can be called phase 1 or phase 1B that you can do with human beings. So basically we mimic the same identical thing that actually we had done you can do in human, but actually done in mice. So the bottom line is that was very important after this screening is that we identify a couple of potential molecules. Actually, I can tell you that there are different kind of very complex kind of metabolism. The metabolism of polyphenol is a very complex mechanism that imply guts, but also imply even liver, as well as many other different metabolic pathways. And what we identify, to make a long story short, we identify, for example, two metabolites. One is actually was called malvidine glucoside, and originally actually was found to cross the blood-brain barrier and also to reach the brain. And the other one that indeed is called like a small phenolic acid, which actually is derived from the polyphenol metabolism that is called free HDA, free hydroxycaffeic acid, and which we decided to take as a potential novel generated potential new compound, and then just to say, well, if we treat the human with a polyphenolic preparation, but then we understand that we have two, though these let's say, this kind of drugs that actually we can call drugs, but actually a natural compound that actually generated by your own body after the digestion, and we use those to test in animal model of stress. So there are several models of stress. In that particular study that actually we will discuss in that particular paper.
So Giulio, for my own understanding, so the diet of a human then creates some of these molecules in the gut, and that's where the microbiome is effective. And then a couple of these molecules that you mentioned have been able to essentially get through the blood-brain barrier, so it can actually have an impact on the brain.
It is a very important anyway, and I apologize, maybe I took a little bit kind of more time, but it's very important to keep this in mind. That yes, when I was saying that the original diet that you can it could be the diet, for example, polyphenolic preparation, could be fruit, can be actually vegetable, leaf vegetables, many other different things actually that actually you can use in your own normal diet. But you need to identify which one are the biologically active compound if you want to really to develop from there a pharmacological strategy.
And you need a microbiome strategy too for it to work, right?
That is correct. But you do not have to assume that all the metabolites that are actually generated through absorption in the gut that eventually are mediated by the microbiome are actually generated and are going to generate anything that is maybe potentially of interest or maybe bioactive or relevant to be investigated. So that is the reason you do first animal study, because the animal study allow you to do the screening. And then eventually you validate this kind of a compound that are part of the microbiome as well, actually one of those from the microbiome. And then you're going to have in separate study, you're going to say, well, now I have found that our body is capable to generate these two very interesting molecules. If I'm going to treat an animal that is depressed or has anxiety, I'm able really to attenuate, and then I'm going to avoid to give them all those foods and all the or a huge amount of or actually prevent maybe some of the dietary recommendation. Basically, actually, to generate a new generation of drugs, but actually natural compound generated by your own body. And the answer is yes. And the microbiome play an important role.
So for my own understanding, Giulio, again, so certain polyphenols, ultimately, mechanistically, it could have a sort of a mediating effect on psychological stress, which you say ultimately has an impact on Alzheimer's and dementia. And so, if I understand this correctly, what you're arguing is that you can actually design a diet that, along with, so you could almost custom design a diet for a human in this way.
It is indeed the second part of the new 5 years of investigation. And the new 5 years investigation is actually after we identified that we can identify the molecule, that we understand the microbiome, that actually we have some of these molecules that can attenuate the cognitive deterioration in model of Alzheimer's disease, and also in part that is played by the microbiome. The next question is that can we define a diet that we can give specifically for example, stress-related mechanism that we know are a major risk factor. And by the way, depression and loneliness and anxiety is one of the top. I would say that there's basically probably one of the top risk factor with diabetes and metabolic syndrome and the acceleration of the disease.
Right.
So yes, we have this actually, we passed to the next level of our investigation. We are going to try to treat subject at increasing concentration, and I will tell you then later on about what are this compound. With increasing concentration of this polyphenolic preparation, that are actually dietary polyphenolic preparation, always based on the idea that our preclinical study suggested that once given to mouse or possibly even in human, they generate bioactive compound, basically effective drugs. We are going to give to human an increasing concentration, increasing amount across the time, and then it's try to see, and then it's try to expose a subject that went through this kind of regimen of treatments. And there will be, and will, of course, will be in blind and also in groups. Also, will receive the real compound, and the other one actually will receive the placebo, and then expose all of them to what is called the trier test, the stress trier test. So, in some way, something that maybe I don't know, many people maybe are aware, or maybe now will ask question: what do you do as a stress to a human that is ethically acceptable? So actually, the stress.
Quick question. So these trials, these are subjects. They don't have a diagnosis of Alzheimer's or any new neurodegenerative diseases, right?
No, absolutely not.
Okay, so
and this is the fundamental part of our study.
Yeah.
Because before, and remember that this is the way our NIH now recommend of how to pursue clinical trial.
But how will the endpoint be in this trial?
Okay, well, guys, what if his hypothesis is correct is that we are going to see that two things, that basically the subject that they receive the treatment for 5 weeks exposed to a trier test that is basically nothing else, that actually to be able to give a presentation that was unexpected in front of an audience that is one of the major stressful conditions in human beings, as you might like, or actually, if you ever heard that before. And if indeed that is the case, we should be able to see that that our treatment arm had some kind of function in terms of attenuation, the stressful responses, attenuation of a cytokine or actually inflammatory responses, but actually the placebo will be stressed, and actually there will be an incremental amount of inflammatory responses that are always associated with the stress that actually we are going to provoke.
Okay, so the goal here is the active arm will be more able to deal with stress compared to the placebo arm. Is that the measurement?
That's correct.
And that measurement, granted, it sort of confirms the mouse model observations, but it doesn't necessarily go into, ultimately the beneficial effects that you hypothesize on the Alzheimer's arena, right?
Well, I mean, the bottom line is you have to think about that you need, we are talking about, just for these studies, we are talking about millions and millions of dollars of investigation. Now, remember that one of the most prescribed drug in Alzheimer's disease are basically antidepressant agent, anti-anxiety agent. And to answer your question, the design, and also as it was designed, you probably carefully read what was the original funding of the grants that actually for which I provided you information. The goal was not to study Alzheimer's disease. That is something that actually was even 5 years before for the study that we had done in 2010 and 2010, 2015. In 2015, in the 2005, 2010, sorry. That is actually was a little bit kind of different story, but the center grant that we have funded right now as a fundamental part to try to understand if we can attenuate immune inflammatory responses in the central nervous system. Now, if we want to get into a little bit more detail about how innate immunity in response to stress may promote some kind of Alzheimer's disease, well, and that will require immediately some other kind of investigation in human. Now, we have done several studies even to imply, actually to understand the mechanism of these inflammatory responses that now we can call inflammasome, that is basically nothing else that part of innate immunity, which is in the brain, is localized in specific inflammatory cells. Now, we discovered and we reported that this mechanism of innate immunity in experimental model of Alzheimer's disease may play an important role in promoting subtle but chronic and eventually long-standing inflammatory responses that may be responsible for further acceleration of degenerative condition. Did we prove that? Yes, we did prove in animal model of this disease. And we know that Alzheimer's disease and stress, most likely they certain kind of a certain stage of the disease are actually remembered as the certain stage of age, because age is one of the major risk factors in Alzheimer's disease. They might eventually meet and at some crossroad, and we'll start asking each other. We will start asking each other, can we attenuate this kind of degenerative inflammatory responses to attenuate the progression of the cognitive deterioration that eventually will lead to some kind of a degenerative condition, irreversible and progressive? Well, the answer in preclinical model is yes.
Okay.
But first.
So let me touch on a couple of other things. One of the other projects is sleep disorder-induced cognitive impairment. Is that something that's ongoing, or what are the findings there?
Okay, that is a completely different ball game, but it's always in some way an extremely interesting kind of factor. When the grant was designed to identify stress-related condition associated to some mechanism that can accelerate age-related degenerative condition, well, in this particular case, could be Parkinson's disease, could be Alzheimer's disease. We actually can entertain all our research in Parkinson as well as form of Parkinsonism and mechanism of proteostasis, where polyphenol metabolites may attenuate some of this pathology, which is actually beyond the scope of this discussion. The idea was always to identify some stressful condition. Now, sleep deprivation and acute sleep deprivation, as well as also acute or actually chronic sleep deprivation, but our work on acute sleep deprivation actually led us to really to understand something extremely phenomenal, that even just 6 hours of acute sleep deprivation, actually a few hours, but then we become chronic, but even a couple hours of sleep deprivation, because I have to done because it's done at different time of the day, just to because of rodents, actually was sufficient enough to then once tested in a behavioral testing for cognitive learning, for example, with the classical what is motor testing. We identified that actually even a short period of time of sleep deprivation actually will lead to cognitive impairment. And we were very interested to try to understand some of the mechanisms through which this polyphenol, again, always starting from this bioavailability of the studies that we have done in mice, and try to see if there was some kind of mechanism that we could actually attenuate or actually, first of all, to reverse the behavior. Indeed, it is something that actually we were able to try to identify. And once we used a technology, actually the wide RNA sequencing technique from animals that were sleep deprived versus animals that actually were actually we call placebo, in the sense that animals that were not actually were left in the cage, basically the control group of mice. Interestingly, what we found that as the same level of how you're going to eventually have a stressful condition that theoretically is now considered to be always related to some kind of associated to some kind of inflammation, is exactly what we found in the brain of sleep-deprived animal. And the interesting part that I now recollect with Alzheimer's disease is that we found that the same molecule, like called the innate immunity in the brain, were actually activated and then were associated, and this is actually a paper that is now currently being reviewed, were associated to certain kind of regulation of circadian rhythm genes that in some way were able to control the daylight and sleep and day-night shift of certain kind of metabolic pathways, hormonal pathways, and was completely disrupted in that particular case. So the bottom line, we associated the inflammation to some of these kind of cascades of events. And one thing that's very important to keep in mind, the message from, again, that is unified to all the discussion that we had just had in the last few minutes, that is that in some way, if you have, for example, jet lag kind of stress, cognitive impairment, all of us experience this kind of a condition, or people for a shift, they work at night, for people working in shift and have some kind of sleep deprivation. All of these kind of things have been very well reported, including disease like a sleep apnea. That is one of the most important things and disease that right now is widely investigated. And we've done already some kind of investigation in part of the human. What we found is that are associated to the activation of the kind of innate immunity inflammasome cascade that actually we found in the later stages, middle, earlier, we can call prodromal stages of people, of subjects that develop a very early stage of cognitive impairment in brain of Alzheimer's disease. So now, are we talking about two completely different things? Well, it may be possible. Are all the Alzheimer's disease associated to sleep deprivation? Well, sleep deprivation is one of the major problems in subject, primarily with later stage of Alzheimer's, not even in cognitive impairment, but there are even subjects in prodromal, even in preclinical stages. Prodromal is basically somebody that is asymptomatic, but still have some neuropathology in the brain. Preclinical is basically somebody that doesn't have anything that actually is supposed to be at risk, because age is the only major risk to develop Alzheimer's disease. So, go ahead.
So let's close with touching on another one, which is your lab is also focused on building a model based on genetic and environmental information to predict cognitive decline in overweight and obese individuals with type 2 diabetes. You want to talk a bit about that? This is called the Look Ahead. study. And so, would it be correct in saying that there is a correlation between overweight and obese individuals with type 2 diabetes and cognitive decline? Number one. And number two, people who are overweight and obese individuals may be pre-diabetic or maybe diabetic through lifestyle changes could potentially slow down the issue that might happen in the cognitive decline arena. Both of those statements are true.
My recommendation is yes, yes, yes. As soon as you reach middle age, but this is something that we should start into education, young people, even in the 30s, in the 40s. We can start doing prevention of Alzheimer's disease at the age of 30.
Yeah.
And one of the fundamental parts, as you said, is yes, can we reduce obesity and attenuate diabetes, and eventually to reduce the incidence or the prevalence, depending upon what kind of population you're going to look, and then reduce the onset of a degenerative condition in the brain? The answer is yes.
Yeah.
Any physician should recommend to anybody past the age of 40 to have a reasonable understanding of what is the destiny that they are creating for themselves, primarily just for something. Remember, something that is related to our nutrition.
Yeah.
Remember that once you're gonna look to yourself on the mirror, and then you're gonna see you're gonna see yourself. You're gonna just say, "This is your brain on food."
That's right.
And I conclude that.
Yeah. That's excellent. Yeah, thanks so much, Giulio, for spending time with me today, and good luck with all this important research that you're doing.
Fantastic, Jill. Thank you so much for having me.
Thank you. Bye.
Take care.